So good afternoon, everyone, and welcome to today's SUMAIR seminar. We're joined by Dr. Igor Chernyavsky, who is the University of Manchester's Presidential Academic Fellow in Applied Mathematics. His research focuses primarily on the mechanics and transport processes in complex biological systems, such as the human placenta. He develops image-based multiscale methods for solving fundamental problems of mathematical physiology within hand's reach of clinical applications. Dr. Chernyavsky, thank you for your time today. We're very excited about your presentation, and I'll now pass over to you.
Thank you very much, Kathy. It's my pleasure, and thank you to Vangelis, and to Pani for inviting me today. And I'm going to tell you something about quite a unique system, the human placenta, and in particular how we can develop techniques and approaches to describe how its structure can be translated into the function. So before I move on, I really have to acknowledge many people I work with, and lots of the results I'll show are done by brilliant postdoctoral fellows which I worked with, Alexander Elish and Philip Pierce. Philip now is a lecturer in UCL, and Alex is in France. And also there is really huge experimental input, most recently, as I'll show you, with the collaborations with the National Synchrotron Facility in the UK, led by Win Toon. And of course, we have brilliant clinicians who help us to understand what this all means. And the supported funder supporting the research are given below. So without further ado, I would like to start by asking how many of you might have heard of the book by Erwin Schrödinger, What is Life? If you haven't seen this book, I highly recommend it. Not only the Schrödinger is famous for foundations of quantum mechanics, but he also had deep insights into the physics of living matter. And indeed, he predicted the DNA before it was discovered as an aperiodic crystal. But what was interesting, that he was talking about different scales and physics of different scales. And in particular, he was talking of chemistry as physics of small scales, astrophysics of as a physics of large scales. And I would argue that biology, to a large extent, is the physics of multiple scales, which are intrinsically linked and cannot be decomposed and decoupled fully. So they have to be studied in the fullness. And the human placenta fascinated people for quite some time, and starting from Leonardo, where he quite faithfully represented the vasculature of the womb. And you can see the placenta connected by the cord here. Later on, then macroscopy started to become more prevalent. There was some confusion, and for example, there is a homunculus where they thought first that people are just small humans which are grown as scaled up to become large fetuses. And more recently, there are really amazing developments where some groups are thinking of making placenta replaced by an artificial placenta, or effectively growing very mature fetuses outside the womb in so-called biobag. And that is no longer science fiction, it's a possibility to maintain the support or life support of the in this case lamb fetus for almost a month outside the body, which this stage of maturity, they're not capable of breathing by lungs. And so this technology is emerging, but they're all united by this cord, by this connection to the mother, and the support is provided by placenta. So I would like to tell you a little bit about the structure of placenta. And placenta is absolutely unique. It's unique not as that it really takes over every function of which later served by adult body. So it's a lung, it's a liver, it's a stomach. It provides all nutrients and also immune support. But also it's quite diverse evolutionary. For example, lung and heart, they're somewhat similar in mice and humans, while placenta are very different. And that is interesting, but on the other hand, placenta is quite readily available for study because it's a clinical waste, and you cannot really study lungs or human hearts in large quantities, but you do have access to human placentas. And therefore, on one hand, trying to understand if we can build representative models, theoretical models, but on the other hand, we have access to experimental system which we can use, at least in part, to inform the structure and to validate the model. So the structure. As I said, it's immensely complex, and until recently, there was not a single technique you can use to characterize the structure of the human placenta because of the immense complexity of the organ. And normally it was a combination of many different modalities. So you use the X-ray, confocal microscopy, some electron microscopy, and it's all been mixed and matched, and typically it's not done within the single organ. And very recently, almost hot off press, this looks like a stadium, but it's not. It's a diamond light source. It's a huge synchrotron, which accelerates electrons to very high speeds, and it has a really enormous luminosity, high flux. Certainly, thousands and thousands stronger than you can achieve in a lab in a typical standard microscope. And with that, you can start getting the multiscale structure in one modality for the same sample. So I will show you a movie where you can see a reasonably large sample, certainly for tissue standards, which about 8 millimeters cubed. And you can appreciate that it's immensely complex. It has different types of vasculature, multiple domains. And what you'll see as we zoom in a few moments, that we can start decomposing these domains into the maternal, fetal domains and the interface. So we take a small region, and we first look at the maternal domain, which is the porous domain. And this is done by automatic, semi-automatic supervised model. So you can see the porous structure of the placenta, which is again very complex. And in between is the fetal, embedded fetal structure, which is a blue, capillaries and vessels. And that's the interface between the mother and the baby. Again, the fetal part, just on its own, of a huge variety of scales. And as we zoom in further, and I'll try to pause here just briefly, you can see the actual finest scale in the placenta, which is the true interface for many solutes such as oxygen, where you can see these capillaries surrounded by the tissue, and outside is the maternal domain with maternal blood. So the two blood never mix, but the solutes, the water and particular compounds can cross the barrier. So the question is, how can we link all this structure to the function? So let me talk a little bit. So you'll see just some, so that's a scale of human hair. So it's really fine. And the whole placenta is about 10 centimeters. So there is really over 4 orders of magnitudes in space, at least. And here it's somewhat summarizes at least the fetal side of the placenta. You can see the whole vascular tree from the umbilical cord branching and branching into finer levels. And then if you take cross-section, these branches actually packed in the tissue, forming the porous medium. And inside this solid face of the spore are these capillaries, which provide the site of exchange. If you zoom even further, you can think of the really interface, so they can see for scale of the erythrocyte, the red blood cell, on the fetal side, so this fetal capillary, and outside is maternal space, so it will be similar maternal blood outside. They don't cross, of course, unless in extreme pathology, and it's very, very thin, and as often the case for membranes, it's reach of all kind of functional transporters, depending on energy supply. But in this talk, I would like to focus on the simplest possible means of transport, which is the passive transport, or transport which is driven by the gradient of solute itself, not requiring external energy supply. So that will be gases, some compounds which are either endogenous or might be some simple chemicals which are used as drugs, and might be glucose, but nothing more complex than that. And even with that, you'll see that it's a very non-trivial problem. Now, before we move on deeper in the placenta, it's always helpful to think of analogies. And one analogy here could be the blood-brain barrier. For example, if we think of many compounds which many pharmaceutical companies worry about, they tend to define the permeability across the brain barrier as a function of how it is soluble in oil relative to water. So it's how easy for it to cross the membrane. And typically you'd expect that if it's well soluble, also partition coefficient is high, then permeability will scale proportionally. But there are some outliers, as you can see, which cross less than expected or better than expected. And the reason for that, because we have transporters. As I said, one that facilitate the transport and one which actually actively pump out to protect against possible toxicity. So what will be nice is if we can start to put a different chemistry and different biochemistry combined with the physics and structure to try to predict the performance of the human placenta. Because certainly it's not a single purpose organ. It really has to balance many demands. And in terms of models, if you think of the physical models, then originally it was a black box, which only was treated as things come in and things come out like oxygen, without any appreciation of the structure, which changed about 10 years ago when they became more simplified, idealized models of porous-like flows in the placenta. And very recently, literally in the last 5 years, there's an explosion of image-based models, partially enabled by the rich data coming from different modalities. And most recent example is the synchrotron I showed. And again, that's immensely complex and it's highly variable, typically low number of samples per organ, low number of different groups of patients and so on. So even though it's data rich, it's very hard to make possible clinical conclusions from that numerical experiments. So the time perhaps is ripe to go back and revisit the black box, but enlightened by all these complex arrangements, and try to systematically upscale and learn key parameters from the microstructure at different levels. So that's more or less the program for what I'm going to present, is how we can revisit as simplified reduced models like you see on the left, but rather having a totally black box, have somewhat a gray box, which has at least some control on the underlying structural parameters at the organ scale. So first, let's look at the maternal side of transport. So that's, which is the porous medium, which is filled with maternal blood. And again, if you take a snapshot, so that's the green is the fetal capillaries surrounded by the tissue. So there's a monolayer or fused layer of cells, which is the orange, and black is the maternal space filled with blood in general. So how can we model a transport through such a medium, ignoring the fetal compartment for the time being, seen it as some prescribed sink for solute? Well, one can take an engineering approach and think of an effective medium composed of some structure and try to upscale the resistance to flow and transport parameters. And that's typically done in porous media, where we take a detailed models for flow at microscale and upscale it to get the average properties, average flow rates governed by, for example, Darcy flow. And the link between these scales will be given by some parameters, such as the permeability, which in general will be tensor form, totally defined by geometry. So once we know what's the microscopic geometry and identify some representative scale for extracting this averaged quantity, we can predict the resistance to flow and also quantify possible anisotropy. Now, I'll come back to that later, to what is the right scale to extract this information. But for now, let's say we know, for example, some estimate of resistance. So we can say that flow is quite slow generally, so Reynolds number is not high, and you can appreciate that it's quite dense medium. So you see, for example, a spiral artery, which is the maternal supply to the placenta, coming from that side. So the bottom is maternal part, top is the fetal part, and you can see here one tree, which is we call the placentone, somewhat a functional unit in the placenta. So we want to model the transport and flow of maternal side of the placenta supplied by the arteries and drained by veins from the mother perspective. If we look at the Peckel number, which quantifies how fast is the flow relative to diffusion, then we see that it's certainly quite dominant at the scale of this unit, of this placentone. So we just take a steady Darcy flow, which is just saying that the flow proportional to the pressure gradient locally, and advective transport, so ignore the diffusion, in a hemispherical approximation to the placentone. So we think that the flow and solute supplies in the middle by point source and drained by point sinks. And let's say there applied no flux condition at the hemispherical boundary. Well, there are two parameters which govern the problem. One is the uptake parameter, or Duhamel number, which compares the strength of the extraction of solute by these capillaries inside this porous medium relative to the flux which you provide. And also how close is the source to the sink, because they will possibly create a shortcut if they're very close. Well, that might look like a spherical cow in a vacuum, indeed, but what I will show that even the simplest possible model is useful because it provides some transparent insights into the kind of key physics of the exchange. And indeed, we can get a closed form solution for the flow and pressure distribution and the corresponding distribution of solute, assuming a particular type of kinetics, for example, a linear uptake characterized by this Duhamel number. And we can vary the parameters and see many transport regimes, which I wouldn't go today, but there will be references which I'm happy to discuss further if you're interested. But that's a conceptual model, and let's see if it makes any sense. And to validate this, we can look in literature, which is quite far back into mid-20th century, where people used X-ray images on monkeys and even, unfortunately, on patients because they wasn't aware of danger of X-rays in America and in Germany. And they observed, so once they inject a particular contrast agent into the maternal circulation, and then they see the serial photographs of projection on the plane, they saw the rings forming and then expanding laterally. And they was puzzled by these rings, and they was speculating what could it mean. Does it mean a particular structure within the placenta will have some cavity-like appearance? And what we wanted to do is to see if we can reproduce this with our simplest possible model. So if we introduce this bolus, the finite volume of this contrast agent coming from the artery, which will be positioned in the middle, and then emanating and forming a shell, which then expands and becomes thinner, then we can project X-ray from the top, which will be then just proportional to the thickness of the shell in a vertical direction. So it's shown by the red line. So it's thickest from the vertical projection in the boundaries and thinnest close to the center. And if you look at this projection of thickness isolines, you can immediately see that they form a ring which expands towards the veins. So if the central artery is the point source, then these two are point sinks, and you can see that this ring forms and expands, and you can plot the area as a function of time, which agrees qualitatively with what they observed in experiments. So we don't need to assume any structural heterogeneity in the porous medium to reproduce the smoke rings. So that was helpful. And now we can start to think about how the structure could affect the function, in this respect, the net uptake from the mother to the fetus. And if we define the net uptake as the amount of solute being delivered by the artery minus what came back to the vein, not consumed by the placenta, then we can see how it varies according to the density of the tissue, the volume fraction of the fetal part of the placenta. And it was somewhat intuitive that it's very, very dense tissue, then the flow will be really slow, so there'll be slow perfusion, and therefore there will be a small net uptake of solute per unit time. On the other extreme, if it's extremely sparse, even though it's really easy to deliver flows through the placenta because it shows no resistance, but there is effectively no surface area available for exchange. So again, you drop to really low levels, and in between you'll get a maximum, local maximum, which suggests that there's some optimal fraction of this tissue, which is called villi, relative to the total volume. And this will depend on type of solute you have in mind, depending on kinetics of the solute. But still, of course, it will exist. So that's just to show that even the simplest model can suggest that, and we can then compare that to real densities, and we show that they have some comparison. But again, it's a matter of parameterization. So what I would like now to spend a little bit more time, because I already see the halfway through, is how we can learn from images what is the relevant scale for extracting this information. Because it's a highly heterogeneous organ, many different solutes. So what is the right scale? How we can systematically identify that? So we can take a section through the tissue. So you can see blue is a tissue stain. And if we just focus on tissue versus not tissue, again, as a volume fraction or error fraction quantity, we can say how our error fraction varies as we change the domain of interest. So the region of interest for extracting the property, such as the average volume fraction or density. And if we have very small window, there will be wild fluctuations, of course, depending where we fall. And then it approaches somewhat the macroscopic homogeneity. But that's still very qualitative. And thanks to recent synchrotron data, we can make it much more quantitative, much more rigorous by taking a large cube of tissue and seeing how the error fraction varies over the different sections of this cube, and then thinking of volume fraction as a function of the subcubes randomly positioned within the sample. And again, as expected, the fluctuations will diminish as you increase the region of interest. And even more quantitatively, we can think about correlations, because it's not a totally random structure. It has a packing associated with the trees and branches. So we can think about the correlation functions and how the relative variability, both, for example, for volume fraction or for specific surface area, which will be relevant for exchange, varies with the region of interest. And we can see immediately that there will be transition where it becomes uncorrelated. So we can somewhat define a mesoscale where it's not yet the size of the organ, but it's still large enough not to worry too much about local fluctuations in the morphometry, in the parameters which define the structure. And even this, again, I wouldn't go into details, but if you instead ask me, we can even get a scaling of how much the error drop as we increase the size of this sampling tissue, because in the limit and asymptotically, we can show that it scales inversely proportionally to the square root of the volume for sufficiently large volumes. So that just to give us some hint what is the right scale, so of the order of hundreds of microns as opposed to millimeters, which is helpful. And now if we take a large cube, we can start quantifying it. So for example, we can decompose the fetal part and the maternal part and get the corresponding networks, like the porous networks or the vascular networks. Or we can run simple flow simulations and try to understand anisotropy and effective properties. Or we can quantify the statistics of these networks. But that's, again, very recent results, and they're still ongoing. But what I wanted to highlight, that this is really open opportunity to start directly correlating the fetal and maternal domains of the placenta, which before was not possible at all. And in the second part of this talk, I'll focus on the fetal domain and tell again how we can effectively extract some information from rather complex structures. Just to summarize before I move on, The maternal transport is highly complex, and there's a variety of possible transport regimes, and I didn't spend time talking about all of them, but you can appreciate that depending on how diffusible is the solute, you might have one way or the other to describe it. But what's most important is before we move into any of this, we need to sort out the uncertainty in the micromorphometry, and only within last few years this is becoming possible to identify what is the mesoscale for extracting the effective properties. Okay, are there any questions at this point? If not, I'll move on, but please feel free to ask.
May I ask you, Igor? Yeah, since you stopped now anyway. So I'm just wondering here about the uncertainty quantification, as you say here. I mean, how exactly do you study this? Are you using specific statistical, let's say, approaches for that?
Well, this was the more or less the approaches we took on this slide, for example, where we can effectively identify how fluctuations in the parameters, such as the fraction or error, specific area fraction, vary, and so that autocorrelation function really kind of a key here to identify lots of the statistical properties. Mm-hmm. So that's at the structure level. At the transport level, we can.
Since sorry for the interruption, since you are now in this slide, I'm just wondering what is how you define this 2-point correlation function? Which correlation function is this?
That's the function which effectively defines how likely effectively a segment of a given length to fall in the same phase in the tissue or in the void.
Okay.
So that's effectively saying how likely, if I fix the position, if it's isotropic and homogeneous, in this case it does not depend on the orientation or on location. If in this case, if I fix the position, how likely a given distance from this position I will encounter one phase or the other. So are there any correlation in predicting the phase?
I see. So it's a measure of the local heterogeneity, let's say.
Yes, that's right. Yeah.
And you have 2 phases, right?
Yes, in this case we only worry about 2 phases, yes, so as a porous medium. But you're right that in principle, of course, it becomes much more complex when we think of all phases at once, which at least we have 3 phases: the tissue, the maternal domain, and the fetal domain.
It's okay. I'm just trying to understand this like an image-based, let's say, analysis that we're doing.
But that's a very powerful approach, because if we identify this length scale, we can then take much simpler approach, because for large enough distances, the errors no longer care about microstructure. They will be just a factor, and they only care about the volume.
Okay, thanks.
So I will come back to this uncertainty a bit later, but let me move to the fetal side of this story. So if we now jump all the way from the porous side to the fetal side, then again you can see that interface, which you've seen a couple of times. So outside the maternal domain, inside the fetal blood, and in between is the barrier. And now I want to focus on the transport within the fetal network. And we can extract these images. This time it was from confocal microscopy, and we can get the topology of this network. And of course we can run simulations, for example, using standard CFD approaches. But that's rather expensive, and all the networks doesn't look like each. They all doesn't look alike. So again, the question is, can we get some generic universal principles which we can use to predict the function? So let's say we prescribe a Stokes flow for a fixed pressure drop. We use this time advective diffusive transport in the network, and also important to note that there will be an advective factor called the facilitation factor, which is due to red blood cells having capacity to transport oxygen much more than the plasma, and also for other gases such as CO2 and unfortunately carbon monoxide, which is a toxic poison. So that can be quite significant. So for some solutes like say ethanol, or for example, there's no facilitation, it's just like water. But for oxygen or for carbon monoxide, it can be as large as 10 to the 4. So it's a huge variation in the kind of boost, how much you can be propelled due to the being bound to red blood cell. And on the other hand, you can think about diffusive flux across the barrier, across the interface. So normally if I come in person, I'll bring with you this 3D printed network, but you can appreciate it at the distance. So that's again a reconstructed geometry for one sample. And we want to define the flux across this barrier from the maternal to the fetal side. And that can be entirely prescribed by effective length scale, which in a simplistic way, if it will be just, for example, a sphere or a cylinder, we'll just measure the ratio of the area to the thickness. So the thinner it is, the larger the surface area, the larger flux you can deliver, which is more kind of the way of thinking traditionally employed in clinical physiology literature. But of course, that is not a sphere, as you can appreciate, and that's it's a length scale which can be estimated numerically for a prescribed gradient and prescribed diffusivity. But it's just purely fully defined geometrically. That's just a length scale. For every given sample, you get a length scale associated with the diffusive flux. And the good news is that we'll show that only 3 parameters are sufficient to comprehensively describe kind of key parameters of transport. And that are the resistance of the network, which is a traditional kind of resistance to flow to the Stokes flow, or if you have blood, non-linear, non-Newtonian flow. That parameter, which is the diffusive exchange capacity, which is effectively just length scale times the diffusion and the concentration gradient, and for comparison, a length scale network, for example, defined as a total length of all capillaries or the longest path from inlet to outlet. So if we take these parameters, we can compose some dimensionless quantities. And in fact, 2 of them are useful. One, which is called the diffusive barrier capacity, which is a ratio of the diffusive length scale to the size of the network, and also the ratio of diffusivity of the solute in tissue and in plasma, which of course will be, say, 1 for simple solutes like oxygen, but for larger molecules like glucose, it's not the case, because it takes much longer to traverse the tissue than it is to travel in the plasma. And even more useful parameter is the yet another Damköhler number, which compares the advective transport to the barrier transport. And again, it compares the advective flux across the barrier to the diffusive flux across the barrier, which will depend on the same length scale for the barrier. And delta P over R is just the flow rate in the capillary network. And again, it will depend on the diffusion across the barrier. So you can see that they're red and blue here, and they to highlight the different contributions. So the red is a geometry and blue is the physics or biochemistry. So for example, what I want you to keep in mind that this ratio of B over D, it effectively shows you something about the chemical features. So how much it binds to red blood cells, how easy it is to cross the barrier in terms of its transporter features, while L is just a fixed parameter for a given sample. So blue parameters can vary, while red are fixed for a given placenta or for a given part of the placenta. And if we plot in a naive kind of raw quantities, the uptake rate numerically for CFD, which is very expensive, this very complex 3D structures, in 4 different networks from the same placenta, and varying the flow rate in the network, you can see that of course it approaches some maximum capacity as the flow rate is very fast in the network for fixed supply of solute outside. But it's quite variable. All these geometries are very different, and the uptake varies almost within an order of magnitude, both for low and high flows. So the question is the way to simplify to get a universal description. And of course, the reason I'm presenting it to you today, the answer is yes. And these two parameters help to achieve that. So if we define the same picture, but in terms of the Damköhler number, which is the advective capacity, and a scaled maximum flux, which will be, again, fully defined by this length scale, we see that all this complexity disappears and it collapses on quite a universal low, which of course reaches maximum capacity and scales more or less linearly, because it's more or less kind of Poisson-like flow for very slow flow. And in between there, of course, will be some transition. And even more helpfully, we can predict the shape almost precisely, quite accurately, certainly in the limits, using asymptotic approaches. So we can think of a limit when it becomes constrained by the barrier, which is that side, or it constrained by the flow in the network, which is that side. And if we get these composite asymptotics, we can see that it's actually doing a very good job with no more than like 30% difference between the numerics, which is for the biological structure is kind of quite nice. And it certainly goes from spending 14 hours per computation to a few seconds. And of course, if we want to scale up for a whole organ, that's the only way forward. Now, if we start to put it all in the context of different solutes, because we can compare oxygen with glucose and so on, we can get a quite comprehensive map of diffusive capacity in terms of how easy it is to cross the barrier versus how quickly it gets cleared away by network in terms of the advection in the capillary network. And we can start adding the solutes on the advective capacity, for example, for oxygen somewhere in the middle. So diffusion limited means that it's hard to cross the barrier, so it's limited by diffusion in the tissue, while flow limited, it's easy to cross, but the flow can be improved and bring you high flux. So you can see the spread is quite large. Again, it's 4 orders of magnitudes. And even the same story happens if you start to think about easiness to cross the barrier, because it's a 2 dimensions to it. And that's, again, not traditionally appreciated, that you have 2 sides to the story. One is the flow related and one is the structure related, and they are interlinked. They cannot really be separated. So if you want to in principle, deliver more solute, you need to move diagonally here rather than just one direction. For example, you can see here that the ethanol or urea, they have a similar, for a fixed kind of diffusive capacity, they have similar flux if you move kind of across this isoline, but they live in a very different regimes. It's one, for example, observation, and another one is that on this side where you have diffusion limited transport, 2 solutes, they're quite different. So fructose, it doesn't bind to red blood cells, but it's so slow to cross that it's really limited by the barrier. While carbon monoxide, which is a poison, it's really easy crosses the barrier, but it binds so dramatically to red blood cells that it effectively cleared immediately and never kind of diffuses outside of them. So both of them, for different reasons, become limited by the barrier thickness, which is again not obvious until you identify the parameters. So the power of this approach is that rather than thinking of understanding the function for a given solute, we identify the parameter which captures at least a class of solutes, a reasonably wide class, at once. And then next step is possibly asking questions how these features where we are in placentas for normal and abnormal pregnancies, which is kind of the program for future research. But at least now we know what to look for. And I'm aware of the time I'm getting close to the end, so I'll skip some details. So this is just to say that we looked into the robustness of this approach at the kind of subnetwork level. And we looked, for example, is prescribing more rheology relevant models. So, for example, applying some hematocrit dependence on the radius and seeing how it will affect the uptake. And we show that it really have a minor effect and the network is quite robust. And also, quite interestingly, in this transition where you are not very flow limited and not extremely diffusion limited, there can be a subnetwork non-monotonic behavior. You not necessarily go just from low uptake at 0 flows to high. At high flows, you can actually go down and up like this, or up and down. So there can be some kind of donor-recipient effect if you have arrangement where one branch and another branch have counter current flow, then they can locally serve as a donor recipient. But on the scale of the whole network, these will average out, and we wouldn't see that. So for large enough networks, that will be monotonic still, but it's still interesting. And just a few words about metabolism, because so far we just talked about getting stuff from one side to the other, and just wanted to know how quickly we can get that for different solutes. But in fact, placenta, of course, is a living, breathing organ. And we have, that comes with the power of collaborations, and we have quite a complex machinery where we can maintain placenta under physiological conditions outside the body, which is called ex vivo placental perfusion. So that's quite a small proportion of the actual equipment needed. So that's oxygenated, oxygen is delivered from both networks, from maternal and fetal side. And we can measure with the optical sensors the oxygen as a function of time or position. And what we can do, so you can see the needle for reference. And sorry, if there's a bit of medical pictures, so I hope you're not too sensitive. What we can do is we can measure, for example, metabolism in the human placenta this way. So we can stick a needle and switch off oxygen and see how quickly it drops in recording. So that's the maternal supply, the below is the fetal supply of oxygen, and then we stick a probe, which is the area, and see how we drop as a function of time. And if we think of a simple Michaelis-Menten-like description, we can extract the kinetic parameters, like a maximum metabolic rate and the half saturation concentration. And then we would like to try and put it in the context of the other organs and tissues. So, for example, we know brain really depends on oxygen, and it has very high metabolism, and so is the heart. But, for example, liver might not necessarily be so metabolizing or not, and it's not clear. And what is interesting, the placenta does not seem to fall within the very highly metabolic tissues, so it's relatively modest from what it seems. But what is more interesting, but again, that's preliminary and very pilot, is that it looks like that this transition to the maximum metabolism is somewhat slower in placenta than in other tissues. So it's rather interesting. So if the placenta is unique in its metabolic behavior, so that remains to be seen. But what we can do, at least for now, using this, say, maximum metabolic rate as a guideline, we can then start getting not a two-dimensional behavior for transport, but three-dimensional. So that will be the advection, barrier diffusion, and the metabolism in the barrier or in the tissue. And indeed, if we have a weak metabolism, then things will get through without much of a hindrance. And for strong metabolism, you might not even get anything across because it will be consumed by placenta. And you can get very patchy distribution of solute on the network. And that means that we have another parameter, and we can define it again by appropriate dimensionless analysis. And this will be comparing the metabolic uptake in the tissue to the diffusion and appropriate geometric parameters. And we can compute it for different solutes. We can look at the oxygen, the glucose, fructose, and for many of them it's not significant, which is probably somewhat expected because you don't want placenta to stand too much in the way of getting oxygen. But for fructose, depending on the baseline concentration in the maternal blood plasma, you can actually get a reasonably high value of this parameter. So then it's much larger than 1. That means that metabolism is significant. And so you can say that then mother drinks lots of fuzzy drinks, then the placenta can metabolize quite significant proportion of the fructose, which can or cannot medical implications. And we can look at these kind of dimension. As I said, let's look only at the projection, so not look at all three at once, but only look at the metabolism versus the dump kilo number. And if I am a solute which is, say, diffusion limited, so it's kind of really slow to cross a barrier, and I don't have any much metabolism, then I am sitting here. And as I increase metabolism for, let's say, for fructose, I suddenly cross into the flow limited regime, which is not quite intuitive. So why is that happening? So metabolism in some sense makes flow more important. And to get the hand of this, just briefly, slightly hand wavy intuitively, but we have a much more rigorous way to confirm that. is the reason because there are different mechanisms driving the diffusion limited and flow limited exchange. In diffusion limited, everything is really dependent on the gradients in the barrier. So our flux in the network will be governed by this gradient. While in flow limited, because it's really relatively quick to cross compared to the flow, then it has time to adjust, and the flux will be just proportional to the Poiseuille flux. So flow times the concentration. So it's reading off a concentration which you provide at the inner boundary, at the boundary from the fetal point of view. So as soon as you add some metabolism, the gradient will diminish slower than the average concentration in these regimes, which explains why you get this flow kind of enhancement, importance of flow in the presence of metabolism. And again, as I said, we showed it much more rigorously using the WKB approximations, comparing the exponential contributions from these fluxes. And indeed, the flow is a limiting factor for sufficiently strong tissue metabolism, which is not expected. Okay, so that was a slightly quick walk through the fetal side of the story. And what we found there is that, surprisingly, despite the enormous complexity of structure, there are relatively few parameters which can be directly estimated from images, which accurately predict, at least to the within 30% of error, the flux for a wide variety of solutes. And in fact, I believe that will be useful not just for placenta, which we developed this and validated this on, but in many other complex microvascular systems like brain, kidney, liver, spleen, and so on. And once we know what to look for, we can also ask questions of which of these parameters are affected in pathologies and what can be done to bring them back to the normal condition. So with it, I'll finish, and there are some references here, and I'm happy to answer any questions.
Panayota, you can, I think you can unmute yourself and please go ahead and ask your question.
Hi, Igor. Thanks a lot for, as always, a very, very stimulating talk. So I've got plenty of questions, but I'll start with a few. So, my first question is the structure of the maternal side. Do you have any vasculature at all from of the maternal side, or is it just plain tissue?
It's a good question. I think the actual uniqueness of placenta is that it's really probably the only vasculature in the body which has another structured classical large artery, small artery, arteriole, capillary, small venule, large vein, and so on. It's literally a disordered porous medium flow. So the maternal side does not have organized macrovasculature in the traditional sense. So it's literally a pool of maternal blood which supplies nutrients. So it's truly porous medium flow of non-Newtonian suspension.
Well, that's very interesting. And just so just cells bathing in this fluid in the blood.
Sorry?
And so it's just cells that are bathed in the maternal blood. So the maternal side of things.
You ask about the what we're predicting in the first part, or I'm not sure.
No, I'm just asking. So so there's no microvasculature. So we can picture it as just the hemisphere picture that you showed at the beginning. So there's the fetal part, which is that vasculature that we can see branching, and the part of the maternal side consists of cells that are bathed in the maternal blood.
Yeah.
That comes in and out.
Yeah, that's true. So what if you probably this might be helpful to look at. So that summary. So if you think of that picture where the brown are the interface of the porous medium. So the outside is the maternal blood, inside in the green bits are the fetal blood, and then between is the tissue. And that literally, you can, if you ignore the fetal part, which we did in the first part, just focus on the maternal flow, then it's just a porous medium flow. So you percolate in between that dense structures.
Okay, thank you. And one more question. So what things? So I mean, it's amazing how you what things you can capture with simplified models. What are the things that cannot be captured due to heterogeneity and going to the details of what's happening? Are there any phenomena important also medically that have to be resolved? You have to go to smaller scales to capture.
It's a very good question, and I think to properly answer it, we need to first get these full detailed microscopic models to know when they cannot be upscaled. And that's certainly something we're interested in, and at least starting slowly to move in this direction. But one certainly important aspect which is not possible to directly describe with a highly simplified model is once we go into more complex compounds. So when the transporters are involved, we certainly have to have a model which works at the membrane scale, and then the distribution of these transporters is likely to be heterogeneous. So how, and the good question is, where would you expect these transporters to be relative to the supply? Because they often thought that once you are limited by the diffusion, then it's really not even the glucose, but something which really slow to cross, like amino acids. Then people often say that flow is irrelevant, but it might not be true, because it depends, can you actually get to this transport site at all? Because that will be determined by the flow patterns. So if your transporter is positioned in a place where not much flow coming to from the maternal perspective, then it doesn't matter if it's fast or slow. So understanding how the flow patterns and distribution of kind of molecular level transporters are correlated is, I think, certainly will require very high resolved models, which I don't think even started to be formulated at this stage. And the second aspect is, I was saying a couple of times that blood is a non-Newtonian suspension, and so far, we and many others just looked at it as effectively Newtonian flow, but with a different viscosity, and we just sometimes varying viscosity depending on capillary size and density of red blood cells characterized by hematocrit and so on. But in the porous medium, there are no frameworks to really describe and predict the flow of suspension. Even in regular porous medium, it's hard, but in irregular porous medium, flow of red blood cells, which is non-Newtonian suspension, is very, very non-trivial. So, and this is another question of at what point we can ignore possible localizations and local blockages and distributions of, because its transport is very much correlated with the red blood cells, both from the flow point of view and from the transport, from the exchange point of view. So if you have areas which are effectively devoid of red blood cells, there will be no exchange for oxygen. So understanding suspension flow in the porous medium is another aspect where microscopic models are extremely important. And this is exactly what we are doing starting from January this year.
Thank you. So a quick follow-up question on this last part. What are the ratios of, so what's the difference in size between the size of a red blood cell and the size of that kind of the grains, let's say, of your porous medium?
It's another very good question, and I don't think there is a kind of single answer to that. So certainly red blood cells, they are about 7 microns or so less than 10 microns. And in principle, we might have gaps which are comparable to that size, or we can have gaps which are much wider than that. So that's a feature of placenta, that it's highly heterogeneous, that it's not really uniformly sparse or uniformly dense. So that's the reason why I feel that understanding micro mechanics of blood flow as a non-Newtonian fluid in a porous medium will help answer. So I think the answer will be there will be regions where the occlusions and jams might become relevant, or at least transiently, and there will be regions where you can treat it as effectively a large vessel, but with a non-linear kind of non-straight, very highly tortuous shapes.
Thank you.
Yeah, if I may ask also, Igor, actually, yeah, just to, let's say, to continue a follow-up question. Actually, yes, before going to the molecular level, I think it's of interesting to compare with, for example, classical CFD of the red blood flow, because there are some simulations, even, let's say, in the mesoscopic or in the Stokes-like level. And I think in some cases they try to use even non-Newtonian, to take into account non-Newtonian effects. Even recently, I think there are also some works using machine learning type of approaches for that. So I was just wondering if you compare, let's say, your data with such CFD, let's say, based simulations, is this something that is doable, or it's?
Yeah, I think again, I'm not sure I can answer that question now. So the plan is to try that, exactly what you're suggesting, is to drill it. What we did is with this, we did CFD of Newtonian flows, which was hard enough. And the plan certainly is to try to see directly if we can flow suspension coupled with the oxygen, for example, or some simple diffusive solute, mobile solute, how much the discrete nature of the flow will affect these predictions. So what we did is somewhat indirectly is we looked at the one-dimensional approximation of this network, and if we use rheology in these networks, which is possible because people did some parameterization for rheology in one-dimensional networks like capillaries, so it's a pris and second. So there is a constitutive empirical laws saying how viscosity depends on local diameter and hematocrit. So we put it in, and as I showed, it didn't seem to make much difference, but it was certainly in a one-dimensional, highly simplified. So is that still be true in a fully three-dimensional geometry? We don't know. So but I think it's possible to do it in a one network, but it's not possible to do it at scale, because just simulation of suspension is even more expensive than simulating the transport.
Yeah, no, definitely these are expensive simulations for sure. So
I think it's feasible on reasonably small network.
So are you using, let's say, large-scale infrastructure for such?
Yeah, so our group personally doesn't do kind of that type of kind of computational rheology, but we have great collaborators in Edinburgh, Tim Kruger and Miguel Bernabeu, and they use lattice Boltzmann framework on clusters to do these simulations.
Yeah, I remember some years ago in Germany, they also used some such kinds of mesoscopic particle simulations, and it was not lattice-based, lattice Boltzmann or something like that. Gerhard Gomber, if I remember well, and some Japanese group that they were using this. So without a direct, let's say, connection to the microstructure, but using particle models in order to take into account these non-Newtonian effects.
Yes, yeah, there are several groups doing this, and I'm aware that there are certainly there is a really nice paper recently trying to model suspension in a network, in a larger capillary traditional network. So it's becoming feasible. But I think our challenge is to try to combine kind of two types of randomness from the kind of physics point of view, is it transport or flow, and randomness from the structure point of view. So if it's a really regular straight channel, there are certainly lots of interesting effects due to the rheology, but we want to know kind of net effect on the transport in the disordered domain. So what's a net resistance, for example, or what's the localization of these particles would be. And also they're deformable, so it's also quite different.
Yeah,
physics.
Yeah, definitely. Yeah. Okay, and if I may ask a last question, let's say at some point you saw this Solute Exchange Map.
Sorry?
The Solute Exchange Map. The Solute Exchange Map that you mentioned at some point, you referred to that. There was this map where you had this advective versus the diffusion capacity. Do I remember well?
Yes, let me just go to that.
No, it was
Yeah,
not
yeah.
to me, what are these points here? So the points are what? Are there data from observations or?
Yeah, so yeah, sorry, I should have actually explained that. That are these 4 samples, which you can see on the previous slides. So that's actually these four networks. So they all, well, and actually they fall on different sides for reason, because these 4 networks being fixed, because it's a complex experimental procedure, they've been fixed at different pressures, which means that they have different resistances in a sense. So that and it wasn't evident on images, on simple kind of statistics of the dimensions like the radii or lengths, but only once we integrate that, if we get this parameter, we can see that they separate according to low resistance and high resistance networks in a sense.
Okay, and how were they chosen, these such networks? Is it,
I don't think there was a particular framework for choose. I think it was just 4 different samples taken from the same placenta, 2 fixed at 1 pressure and 2 fixed at a different pressure, because they have to be fixed under pressure. It's like a resin-like. So it's a kind of contrast agent.
But I would assume that in principle there should be many different possible choices, right?
Yes, indeed, and that's the reason why we wanted to see if we can get a universal way to describe that, because we cannot keep repeating these detailed simulations for every network.
Okay, thank you very much.
Panayota, do you have a question?
Yes. So I was wondering what are the difference between fructose and glucose that make the metabolism so different?
Well, they're both sugars. Just fructose does not have, I think, well, glucose is kind of critical for survival, and it, I think, has much, well, again, I'm not a biochemist to really answer that properly, but my understanding is that glucose are just, have more efficient transporters than fructose. Partially, I think fructose might be large a little bit. I cannot tell that, but certainly in terms of just transport point of view, both need transporters. They cannot cross by diffusion, but we use this as effective diffusion, so that they are driven by the gradient still, just non-linearly. But they will be characterized by effective diffusivity, and that effective diffusivity will be smaller for fructose than for glucose, because of the fetal transporter properties in the membrane.
It's very interesting that the placenta has I think it has low metabolism.
Well, it's not that it's low; it's that it's kind of relatively less important for the exchange. So it's placenta eats. It's a kind of it still breathes and it needs to get supply of oxygen. What I think I wanted to show is not that it does not metabolize, that its metabolism is not significant from the point of view of exchange. And certainly, depending on the regime, it will be more or less significant. So it's what I'm saying for fructose, it's just large proportion of supplied solute will be consumed possibly than for other solutes. But placenta definitely needs oxygen.
Have you by any chance coupled these things or some other collaborators with PKPD modeling?
Well, that's what was trying for quite some time, not yet, but that's definitely something we keep in mind, because that kind of natural continuation of that direction is we want to see if that kind of combined ex vivo model and in silico model could be useful for predicting toxicity and moving more from just simple solutes to slightly more complex solutes. But there are lots of complications from experimental point of view, but yeah, certainly future lies in the kind of integrated experimental and mathematical models.
Okay, I've got one last question. if there's time. I was just wondering about the morphogenesis of the placenta. I mean, especially, I think it was an artery. The maternal artery that goes in the placenta is also spiral.
Yeah,
that's interesting. And also, like how the placenta kind of forms, because I would expect things to branch out from the maternal side as the placenta grows, but it seems that it's kind of the opposite on how the part of the fetal network kind of goes into the placenta.
That's exactly right. So the placenta, in a sense, is driven by fetus with the permission from mother, more or less. That's how it works. So it's a quite active. Well, that's site which I even didn't touch because that's it's another probably 10 years of development. But we only talked here about the fully developed placenta. In fact, it's quite rapidly developing, and lots of complications start well before it's fully developed. And actually, lots of clinical interest is in trying to get good handle of why is it getting wrong. And some people early, and some people just before the delivery. And you can see there's trees projecting here. They're literally growing, and all the material, all the constituents actually is from the fetal origin. So the only part from the mother is this kind of very much a base or placental bed. So apart from this artery, which you can see here, all the rest is the fetal. So it's actually just the mother supply, gives the access to the nutrients and environment, and fetus just make sure that it's well utilized and without. So it's a very fine balance, but at the same time robust enough for kind of being perturbed in many ways.
Because the placenta forms quite early, isn't it?
Yes, it's formed from much from the very beginning, but it becomes functional only towards the end of the first trimester. So it's not really doing any. So there's not established circulation from maternal arteries before the end of the first trimester, more or less. So it only starts actively delivering blood in the space from the around 12th week of gestation. So indeed it's if you Bernard Shaw had a famous quote that no human bears resemblance to a sapling, to a tree, except for the first 9 months of development.
Yes. Okay, so yeah, this is very interesting. So I presume first it will start forming as the porous space, and then once the kind of the organogenesis of the fetus has moved on, then this fetal tree network will start forming.
Are they doing parallel? So the fetus needs to develop the tree structure and the vascular structure within the tree at the same time as making sure that the maternal bed is becoming kind of receptive and can supply solutes in sufficient quantities, because it's another story that which I didn't touch, that the location and the relative scales of the maternal network is important, which is all below this plane. So if the fetal network is fine, but the maternal network is maldeveloped, then there will be no supply to placenta. And that's part of a lot of complications like the preeclampsia and the hypertension during pregnancy.
And is there any reason why the artery is spiral, that maternal?
Well, it's a very good question. I don't think any have a definitive answer why the spiral artery is spiral. There are lots of different theories, and I don't want to advocate any of them. Part of it could be purely kind of developmental, that it just grows quickly and then just so it's quite dynamic artery. It's not necessarily very static. It could be just a mechanical possible compromise, just to ensure that it can be stretched, or it can in principle also limit the flow if you want to close the flow. It's easier to do it if you have a larger vessel, but not necessarily in space, but in coiled. but I don't think there's a clear answer. But certainly it's known that in pregnancy there should be low resistance vessels, so they're quite dilated, quite remodeled.
Mhm. Okay, thank you very much. Really enjoyed this talk. Thank you.
Well, thank you very much, Paniota. Thank you very much, everyone. My pleasure talking to all of you. I'll stop sharing screen.
Okay, thank you very much, Morgan.
Thank you, Igor.
Yeah.
Thanks for your time, and wishing you all a good afternoon. And hopefully, maybe in the future, we'll be able to host you here in Cyprus. But until then, this was a great presentation. Thank you very much.
Thank you for having me.
Bye. Ciao. Bye. Bye.
Bye.
Bye. Goodbye, everyone.
Bye, thank you.